Center. I want to welcome you all to the October edition of our monthly webinar on infectious disease modeling research. Usually we do this the last Friday of every month at noon Eastern time. I want to give an advance warning that in November, we will be having a slightly different schedule, having the monthly webinar at 1 p.m. Eastern on the 19th, which is not the last Friday, but the third Friday of the month. That will accommodate the Thanksgiving holiday here in the US. So please watch for the announcements in your weekly updates from MIDAS and adjust your calendars accordingly. Today, we have the exciting pleasure of being able to hear from Dr. Madikay Senghore. Dr. Senghore is a postdoctoral fellow at the Center for Communicable Disease Dynamics at Harvard, where he studies genomic epidemiology of pathogens, looking at the impact of clinical interventions like vaccination and antimicrobial therapy on the population of pathogens such as strep, staph, and tuberculosis. So we're going to hear from Dr. Senghore about the infectious disease as seen through transmission factors. Before I hand over to Dr. Senghore, I do want to mention and remind everybody that we do have Spanish interpretation available. So if you hit the button under interpretation at the bottom of the screen, you can get access to that Spanish version of the talk as well. And with that, I'll hand it over to Dr. Senghore. We're looking forward to hearing your talk.
Thank you so much, Harry. And if I can just take this opportunity to thank yourself and Jess and Stephanie and the rest of the MIDAS team for inviting me. It's a great honor to present at this forum. And today I'm going to basically give you a brief synopsis of how my understanding of transmission of infectious diseases has evolved over the years. And I want to start off by, just briefly introducing myself. So I was born in the Gambia and I grew up in the Gambia. And, like most kids who had a parent that was a physician, I sort of grew up aspiring to be a medical doctor. And then in around 2006, I went to do an internship at the MRC unit, the Gambia, which is Medical Research Council unit, the Gambia. And I started being introduced to laboratory research. And I ended up, applying for a scholarship through the MRC to do a bachelor's. And, in the end, I ended up spending the next 13 years of my life at the MRC, where I got trained at bachelor's, master's, PhD, and postdoctoral level. I first did a bachelor's in medicinal chemistry. And then after working for a year, I did a master's in bioinformatics and systems, bioinformatics and systems biology. And that sort of led me to the Senghore Institute, where I really discovered about, that's where I discovered about pathogen genomics. And that was the topic of my PhD thesis, was looking at the genomic epidemiology of pathogens in West Africa. And that sort of led into my postdoc. But I got wary after a while that my work was becoming very niche-centric, in the sense that it was very easy for me to relate to people who understood phylogenetic trees and phylodynamics. But outside of that niche, it was not so much easy to communicate my findings. So I really wanted to develop a more quantitative background where I could incorporate aspects of epidemiology and modeling into my work. And that's what really led me to the Center for Communicable Disease Dynamics, where over the past couple of years, I've been trying to become more integrative of all these different approaches and to be more quantitative. So to give more numerical answers as opposed to just giving generic overviews and illustrations of my work. And of course, you know, my journey would not have been possible, A, without the MRC, and B, without really a lot of very, you know, important contributions from mentors, advisors, collaborators, and I've just highlighted a few of them at the bottom of the slide here. So we get started on, you know, the topic for today. I looked into the dictionary to see, you know, what I would see in terms of the definition of transmission. And in very, very terse language, it says a passage or transfer as of a disease from one individual to another. And in the context of the work that we do, you know, proving that transmission exists between two individuals or two communities is a lot like the job that a lawyer has when they go in court, in the sense that you want to argue the case that you can prove beyond reasonable doubt that either this is likely to have been a transmission event or not. In many cases, it's much easier to rule out transmission than it is to actually confirm transmission. So what really intrigues me about studying the transmission of infectious diseases is that there's really a very wide plethora of questions, methods, and data sets that can be applied to understand transmission. You can use models that, you know, use existing data or simulated data. You can rely on epidemiological studies. You know, you can rely on genomic studies as well, which give you a very high precision characterization of the pathogen that you're interested in. And these questions, they can be specific to population. So for example, if the transmission rate is higher in one country versus another country, why is it this way? Are there behavioral patterns that are making it such, or are there genetic factors underlying this? It could be more precise, where you look at a set of hosts and you want to know whom infected whom, or you want to know where they're getting infected from. You know, it can also be dependent on the pathogen. So airborne pathogens, is this a pathogen that's spread by touch? Is it a pathogen that's spread by a vector? And all of these, you know, things, they give you different considerations in terms of how you model the disease or how you would study genomes from this disease. And that's what really intrigues me. So if I just sort of focus a little bit on genomic transmission studies, which is really my core field, my core skill set is in genomic, particularly microbial genomics. So you start off with a population of interest. And in that population of interest, you have a pathogen that you're interested in. And you have specific questions that you want to ask about that pathogen. So for example, if you're familiar with TB, there have been TB case contact studies, for example, where they look for contacts and they ask, well, is this person more likely to have been transmitted from a close contact, or is it something that was out in the community, for example? So you sequence a bunch of stuff from your population. And there's really two approaches generally. You can either do a mapping approach where you have a reference genome and you map your raw data to it, or you can do an assembly where you try and reconstruct the genome based on the sequences without a reference. The benefit of the assembly is that you don't have reference bias. Reference bias means that when you're mapping something against a reference, you can only compare what is on that reference. So there will be aspects that may be in your data set that are not in there. So for example, if you are studying a population from Africa, and you use a reference from the US, there might be significant genetic difference between the reference and your study population that you might miss some signals. But ultimately, what both of them are trying to do is you're trying to establish a set of genomic loci that are conserved in your database or in your data set, which can serve as a basis for comparison. And you'd summarize that data in a multiple sequence alignment, like you see here, where at every position, for example, you have the corresponding base that is present in the sample that you're interested in. And this can be reconstructed as a phylogenetic tree and visualized, but it can also be studied numerically, where you actually count the number of differences between samples, and then you study that. And that's one of the ways to illustrate that is in a histogram, where you basically say, how many of my comparisons in my data set are 0 SNP difference, or they're identical, for example, or 1 SNP or 2 SNP. And you can see that in most cases, what you would find is a bimodal distribution, where you have a bunch of things that are extremely similar to each other. And then you have other things which are a bit similar to each other, but not quite so similar. And mostly transmission studies focus in this part here, where you have things that are very similar to each other. Now, sometimes you set a cutoff and you use that as a basis for finding transmission, but it's not entirely transmission, because you need to have epidemiological data to prove that these individuals were actually in contact and that it was, that the person who you are proposing is the one who transmitted was actually infected at the time of contact, and other things like this. But generally, focusing in this area here, you can really get a good feel for the dynamics of what is driving transmission in your data set, or in terms of specifically answering your research question. So for today, I'm going to really highlight how we can leverage convenience samples. And by that, I mean these are samples of a given pathogen isolated from a human population that were not necessarily collected for the purpose of studying transmission, but they could give you some meaningful information about the dynamics of transmission. And through this, I'm going to highlight, in one example, how we can understand the population dynamics, and also how we can start to understand the individual dynamics in terms of getting closer to understanding who infected whom, or what the risk factors for transmission are. And finally, I'm going to end up with, just asking the question, what is possible if we do have the ideal data set? If we have an outbreak that is really well sampled, what can we achieve in terms of the genomic data? So I'll launch straight into it. And the first example I want to talk about is a study that we did when I was a PhD student at the MRC. And this study had been started by my supervisor, Martin Antonio, and our postdoc in the group, Brenda, as well as Michonne and a few other people. It was a really, really great team effort that put this study together. And basically in the Gambia, one of the tourist attractions that we had in the Gambia, because that habitat has been destroyed partially now by urban development, but we used to have a monkey park in the Gambia. And it was in the center of the tourist district in the Gambia. So it was surrounded by hotels, and it's also where the nightlife was. So most of the nightclubs in the Gambia are in that area. It's called the Senegambia area. If you've been to Gambia, you most likely know where Senegambia is. And I remember when the first time I gave this talk at the MRC, I started off by talking about, so I said to them, well, we sampled the monkeys in Senegambia, and everybody burst out laughing because they thought I was referring to the young people who go to Senegambia to party at the weekend. So I was like, no, these are actual monkeys that we sampled, not the party animals. But anyway, so in this study, Martin was specifically interested in what the carriage dynamics were in these monkeys. And at the time, we did a lot of Streptococcus pneumonia study in our group. And he was expecting that we would find a lot of pneumo in the monkeys. So they teamed up with some anthropologists, and they were able to trap the monkeys humanely and take nasopharyngeal swabs from them. So some of the monkeys were from the monkey park in Abuko. Some of them were a bit further out from the Abuko Nature Reserve, and some of them were also deeper inland in the Central River region, which is about 2 or 3 hour drive from where we are on the coast. But in a nutshell, what we did was we leveraged a convenience sample, because we had invasive disease samples that were being collected from 2002 continuously to around 2010, which is around when the isolates were being compared. And then we also had a human carriage study that had been done a few years prior in 2004. So we took all of these samples, and first thing we did was to do a genotyping, where we just sequence a few genes, 7 genes, and see what the different genotypes are. And we found some interesting results. Most of the genotypes in the monkeys were novel genotypes that were not present in humans, although we did see a few that were humans. So the question arose really is, are we seeing any evidence of zoonotic or anthropogenic transmission between the human population and the monkeys? And the first thing we did was, build a phylogenetic tree, as you do. And in this phylogenetic tree, we incorporated reference genomes that are highly curated reference genomes that represented the global diversity of Staph aureus. And you can see that there's, quite an appreciable amount of diversity across this tree. But what really struck us was this clade here. So the tips on this tree, the green tips represent human invasive disease samples. The red tips represent human carried samples, and the blue tips represent samples that were collected from the monkeys. So we had most of the monkey samples actually formed a unique clade, which we refer to as clade 2. And we were even concerned about whether or not these samples were Staph aureus, given how uniquely different they were. But we tested that and we found out that they were in fact Staph aureus. So our collaborator on this project, Ed File from the University of Bath, he did some back of the envelope calculations because there's extensive literature about the mutation rates of Staph aureus. And we know that in a given space of time, you can expect to have so many mutations. So you can back calculate how far or how long ago two strains diverged based on the number of SNPs. And we estimated that it's probably about 2,000 years or close to 3,000 years since this clade 2 diverged from human clades. We also saw this group of ST6, which is a common human ST samples in the monkeys, and one ST15. And the ST6 samples appeared to be maybe more divergent, maybe 30 years ago. And this was supported by the fact that within the monkeys, you had some that were divergent by about 200 SNPs, 270 SNPs, which is evidence of long-term diversification within this population. So we concluded that, yes, it is possible to have human strains jump into monkeys. We haven't seen the other. And that was something that we wanted to understand. Why was it that the clade 2 samples were not being found in the humans? Why wasn't it a two-way transmission street? So we looked at the accessory genome. So in a bacterial genome, you have the core genome, and this represents genes that are present in everything, or more or less everything. They're very common. And then you have accessory genes, which are present sometimes and they're absent other times, and they sort of latch onto mobile genetic elements that can transfer huge chunks of DNA with like dozens of genes or hundreds of genes at a time. Sometimes they carry antibiotic resistance. Sometimes they carry even immune evasion. Sometimes they carry things that are related to host specialization. So they really adapt, very active in terms of the evolutionary dynamics of strains. And what we saw was that, clade, in addition to having a very similar core genome, the samples within this clade also had a very similar accessory genome, which was more similar than other things. So we dug a little bit deeper to understand, well, what are actually the factors that are either present or missing? And what we saw was that there were a number of regions, like if you look at this ICE complex, this is an immune evasion complex, which is, something that Staph aureus has evolved to escape the human immune system. And this was absent in almost all of the monkey isolates. And these are other similarly, fitness-related regions that are very common in human-associated staph, but not in these monkey populations. So the evidence of ancient transmission followed by host adaptation, that was the conclusion that we drew, because it suggests that the monkeys, once they became adapted to the monkeys, they lost some of these genomic features that would make them better adapted to infecting humans. And like I said, this was really a very big team effort, and it included collaborators from the UK, from the US, as well as from the Gambia. So I'm going to switch gears a little bit and look at a slightly different perspective here. So what can we learn from longitudinally sampled genomes? So typically, if you have a scenario where you're sampling a large proportion of the population or a large proportion of the people who are infected, you have a better chance of identifying, transmission pairs. But what if you only sample a few people in the population, but you sample them intensively? Now, It's not ideal because, as you see on the right of these slides, you only have a few people. So in this case, we had 21 villages and a maximum of 10 children per village. And then you also don't sample any household or community contacts. And then you end up with quite a large number of genomes that are, in many cases, some of them are pretty similar to each other. But what we did was we leveraged this study. And this was a study that was designed by my colleague Brenda, and it was carried out in a rural area in the Gambia. And the study was done to look at the impact of PCV. So kids were in 3 different villages. Some of them had different levels of exposure to vaccine. And the carriage, this was the pneumococcal vaccine. And the carriage dynamics for pneumococcal in the first year were observed by sampling the kids, every 2 weeks for the first 6 months, and then every 2 months for the second 6 months of life. And this is significant because in the Gambia, over 90% of kids will have been exposed to the pneumococcus within the first year of life. This is pneumococcal carriage, not necessarily disease. So we had on average 16 swabs per infant. And when all of those swabs were cultured, there were about 1,258 that were positive for pneumo, about a 75% positivity rate. So we extracted DNA from these and sent the samples to the Senghore where they were sequenced. We started off, as you would do, looking at phylogenetic trees. And, this is just an example of 2 serotypes out of close to 50, for example, right? And what lured us into looking at transmission was that when we looked at this, right here, for example, we saw that, there were samples that were from different kids, sometimes in the same village, sometimes in a different village, but the samples, the bacteria were almost identical. You had bacteria that were like 2 or 3 SNPs different, and here even more so, where you had, quite a bit of evidence that there is some level of transmission in the community, or at least they're getting it from the same source. So we knew we couldn't really study transmission specifically in terms of transmission pairs because we hadn't sampled enough people. But could we use this data to get an understanding of how the transmission dynamics are in these network of villages, for example, right? So we had to think outside the box. The first thing we had to do was, we had to be selective in terms of what we were comparing. I mean, when you have 1,000 genomes, it's not very helpful if you just build a massive tree of 1,000 genomes because you can't really see anything. And then also you're going to have a lot of comparisons that are just thousands of SNPs apart because they're completely unrelated lineages. So we used an existing tool to get phylogenetic clusters. It's called PopPunk, and it uses a K-mer-based approach. So it takes small chunks of the genome, compares everything versus everything, and then it takes the things that are more similar and groups them into a lineage based on the whole genome. And instead of doing a mapping approach, we did an assembly approach, and then we used the genes to get a core genome. For each lineage, we got a core genome, and then we looked at the pairwise SNP differences. And then to get the idea of what would be a reasonable range to deem transmission, we used the within host diversity. So we took, every time we saw a strain defined by the serotype and the genotype that was present multiple times in a child, we took all those different ones and then we got the average difference from them to see what was the average difference between strains when they colonized the same kid multiple times. And we used this to sort of understand what the covariates of the transmission dynamics were. First of all, this is the network of villages that we're talking about. The villages, in some cases, you have villages that are close, and then you have others that are sparse, and then you have some that, there's different levels of clustering. And it would have been difficult to do 1v1 comparisons because some villages had so few numbers. So what we did was we categorized the villages. So we said, 5 kilometers, this is about a reasonable distance for, something that you could walk in a day. So we're going to use that as one category, and say 5 to 15, you could probably use a bike, or you could use like a donkey cart or something like that to get between the villages. So it's something you do if you needed to get there, not probably something you do every day. And then 15 plus, we figured, well, you probably need a car or a motorcycle to go like 15 plus kilometers in a day. That's like a journey, right? So this is how we categorize the village distances. So this is the distance separating when we have two kids that we're comparing. How close are the villages and which of these three categories do they fit in? Another thing that we saw quite early on, and my colleague Crispin Chakuza, he did a very nice job of studying the dynamics of carriage duration and also the evolution of the strains over time. And we saw that in many cases, a strain would colonize a child at multiple time points. Sometimes those time points would be, concurrent, so it'd be a continuous stretch, and sometimes there would be a break. So we tried to focus on these two covariates to understand how transmission works. So the first thing we did was we built a Croucher plot. And this is my advisor, Bill, calls it a Croucher plot because he had a student called Nick Croucher who was, quite a pioneer of this method. And he was able to apply this method very creatively. So he calls it a Croucher plot. But basically what this is saying is you have your comparisons in your database. And you're going to basically categorize your comparisons. And it could be, for example, in a classroom, are the comparisons, are the kids both male and female, or female and female, for example? Or, for example, are the comparisons from the same neighborhood? Are they from the same county? It could be any covariate that you're interested in studying. In this case, we wanted to know how it related to the distance. So what you do is you take a window. So for example, you take a window of 0 to 10 SNPs and you ask yourself, well, within this window, what proportion of my case of the samples of the comparisons are from a close distance versus a more far away distance? And you continue sliding across and you keep doing that. And it gives you a picture of how the dynamics are changing over time. And what we see from this is really that, when two kids are from a close village or the same village, they are more likely to share strains that are more closely related in terms of the frequency here, but also if we look at the genetic relatedness as well, if we look at the actual SNP distance, you'll find that comparisons from a close distance are actually more genetically similar as well. So this was a cue for us to understand, to sort of infer that being closely in a close proximity within a rural setting is actually something that could be a driver of transmission. So the next thing we wanted to do was to really home in on this a little bit more and get a bit more insight. So we tried to set a threshold for what we thought could be a reasonable SNP distance for transmission between strains or between pairs. Not necessarily saying that this is transmission, but that we could, let's say a zone, a hot zone that we could say that, well, what happens within this zone will give us a nice idea of what is driving transmission. Whatever is driving these comparisons is likely to be driving transmission in this setting. So what we did was we took every child, we looked at the strains that they had at multiple time points, and then we compared those ones. So it's the same strain in the same child at multiple time points. We looked at the SNP distances between them, and then we took that for every child and every strain, and then we calculated a mean, which was 23 SNPs. So we said that, well, if you have 23 SNPs in one arm and another 23 in the other arm, then 50 is a reasonable threshold to study. And that's what we did. And again, we found some interesting stuff. So I'll just run through these results quickly. The first thing we saw is that if both kids were colonized by the same strain at multiple time points, in other words, if both kids sustained colonization of the strain and they also shared it, meaning that the strain was very similar between the two kids, we found that in most cases, they were more likely to be from villages that were very close together. And when we looked at the SNP distances as well, again, what we found was that the genetic distance as well is very similar for those two strains when they're from the same strain, when they're from the same village, and they also carried over multiple time points. Not so much if it's sustained in one child and transiently carried in another child. Similarly, if it's transient in both kids, then the difference is less pronounced. So what we're seeing is really that if two kids are from a closely related villages, their exposures are very similar. And this could be for a number of reasons. It could be that the parents, maybe they go to the market and work together. It could be that they have older siblings that go to the same school and they have basically the same strains that are circulating, and that these kids might be even getting repeatedly exposed to these strains. So there's a number of, this is again, interesting questions that you could answer using modeling or with better epidata. And if we look at it phylogenetically, this is what you see. You see that clusters of, for example, all these samples are from the same child, and these two villages are quite closely related. They're quite close together. And this is sort of an archetypal example of, a strain that's really coming into a village and it's colonizing multiple kids at multiple time points. And it's also present sparingly in other villages like EE and GG, which are a bit further apart. So when we take that into account and we actually categorize comparisons as being strain sharing or not, this is what we see at the village level. And we see that there is quite a substantial amount of strain sharing that is occurring across these villages. Now, what is striking is that they are more closely related when they're from this same village or close related villages. But notwithstanding, you have some villages like this one, which is really doing quite a lot of strain sharing with everyone. And we know that within this network of villages, there is a village that is a fishing village where a lot of people congregate. And there's another one which is also like a spiritual healing. There's a spiritual healer there. And a lot of people go there as well. So it could be that this is one of those villages that has a lot of traffic. And another interesting modeling question would be, can we relate the dynamics in terms of strain similarity to actual human mobility? Can we basically infer what human mobility would look like based on a network like this? And again, another great team effort. This was including collaborators at the Senghore, a big team at the MRC, and also my current advisor, Bill Hanich. And it was, this project was led by Brenda, who did a brilliant job of coordinating it. And good news is that this paper was just accepted for publication 2 days ago. So it's something to celebrate. So what if we're dealing with an outbreak? So I apologize because this slide breaks the rule of making sure your audience can actually see the slide. I didn't have enough time to engineer it so that the text would be more visible. But what we had was in a neonatal ward in the Gambia, we had a case where between March 2016 and August 2016, the resident doctor in charge, Udwako Komo, she was paying attention to the patterns of infection, and she saw that there were an accumulating number of Burkholderia capacia cases that were happening. And luckily, they were able to isolate these samples and keep them for further investigation. And then a few months later, after they cleared that outbreak, another outbreak occurred, including extended spectrum beta-lactam-resistant Klebsiella pneumonia. So they wanted to understand what was happening with this outbreak. So we were able to do sequencing on both the Burkholderia isolates that occurred before and the Klebsiella pneumonia isolates. And what was apparent from the Burkholderia was that it seemed to be more of an endemic infection. Because as you can see, there are quite a few different, sort of mini clades on this. They're quite similar. I mean, less than 20 SNPs difference. But you can see that there is some sort of evolutionary branching that's happening. And it makes you concerned about the environmental, the presence of this bacteria in the hospital environment. And because they had previous encounter with this strain in 2015, we were able to do some phylogenetic dating. And again, this sort of exemplifies the challenge that I used to have with doing phylogenetic analysis, because you get an output like this, it's informative, but it's very difficult to interpret. And whatever you get here is not entirely actionable. And that's the key word, is actionable. When we looked at the Klebsiella, we found out that it's actually 2 separate introductions, but based on how similar the genomes were in this case, it was more likely that these were more recent introductions. So again, this was a very nice work that I was invited to collaborate on. It was led by colleagues at the MRC, and we collaborated with Stephen Bentley and others, and Professor Beate Kampman and Imperial. But leads me to the final question I want to ask, which is, what if we could actually tell who infected whom? Because in this scenario here, if you look at this phylogenetic tree, for example, all of these kids, the genomes are identical. There's 0 SNP difference between them. So you'd have to rely on some sort of epidemiological data to understand who infected whom. What if we could actually get more from the genome to inform us on who's infecting whom? And for this, I'm going to pivot and talk about something that I've been working more recently with my advisor, Bill. And we've been relying on a mouse model that was designed by our collaborator at the University of Auckland, Susie Wiles, and her postdocs, Hannah and Priyali. And what they did was, using Citrobacter rodentia, which is also referred to as mouse E. coli, because it infects the mouse gut without making the mice sick, they were able to do a transmission chain in mice. So they'd infect one mouse orally and put it in a cage with a second mouse. And when the mouse defecated, the Citrobacter was tagged fluorescently, so the poop would start to luminesce. So you'd know that this mouse has pooped, and when the next mouse ate the poop, it would become infected and it would get transferred to a new cage, and so on and so forth. And they did this for 22 steps. And they did this experiment 10 times. 5 times they fed the mice with water, and 5 times they fed the mice with nalidixic acid and antibiotic. So in order to understand what was happening within the population or within the mouse, we did deep sequencing, deep genome sequencing, in order to capture within host diversity, with a view of leveraging that to understand transmission a bit better. So classically, you do a consensus tree, you find the base that is most common at each position, and you call that, you do your distance matrix, you do your tree, and you look for your epilink. But when you have a scenario where things are very similar, sometimes that doesn't give you a lot of information. Well, what we know is that, infection in a body is not always uniform. It's not always that you have one strain that's in there. Sometimes you have multiple strains. So for example, these 3 samples, what you might have is actually that you have 2 different samples, and then in one of them, or you have 2 different strains in your host. In one of them, you have a GT combination, in the other one, you have a TG combination. And depending on which of the samples is more abundant in the host or is dominant, you'd get a different percentage. And how this translates is that, well, if this is the individual that's infected, and he's got maybe 3 different variants within him, if he transmits or she transmits to the next person, then some of that population will be transferred. In an ideal situation, you have a proportional transfer, so that, everything that was there, if you had 50% of red, you'd have 50% red here as well, for example. Doesn't always happen because it's a biological system, or should I say, rarely ever happens. But nonetheless, you should be able, in most cases, if there was a red here, you'd find at least 1 red there, even if it was more or less common than in the other one. And then all of a sudden now, the relationship between these 3 guys becomes a bit more complicated. Now, we know that this approach works because, our colleague Martin et al. Have published in Microbial Genomics, and they've shown that, well, transmission pairs are actually more likely to share these sites where you have maybe a 50-50 or a 60-40 or a 70-30 relationship between 2 different bases, right? The question for us is that, can we make this a quantifiable and actionable output? So can we say, if let's say a public health official is sitting in an office trying to understand an outbreak, can we give them a metric that says, well, this one is more related to this one, even though there's 0 SNP difference between them? So we built a phylogenetic tree, and then we looked at how these SNPs are actually accumulating. And what you see is that actually, in many cases, things that end up becoming a SNP start off somewhere very minor, and then they grow over time. So the question is, can we leverage this to trace back to see who's more likely to be infected than whom? So we look, for example, to see if having a shared variant was easy to call a transmission. So like in this case, anything that was more than 10% in the host was considered a variant. So at this position, all of these strains have a variant at this position, right? And what you can see is that, well, there is a trend that as you go over more transmission steps, you're more likely to find a variant in the two, if you compare two things, or less likely to find a variant if you compare two things. But was this enough to actually discriminate? So we used a simple Bayes theorem probability model to see if, well, okay, for this strain, which is very rare in my population, is probably about 5%. If I find this strain in two different things, what is the likelihood that they are involved in transmission? And when it's rare, does a good job. As it becomes more common, not so much. What if we ask, well, what is the probability of being in the same cluster, which is basically 5 transmission steps apart from each other? And it gets a little bit better. When we ask about the probability of it being in the same transmission chain, again, it does a little bit better as well. But if we know that these are useful markers, why is it that when we apply it to everything, it's not giving us as much information as we hope for? And the reason is because we are oversimplifying the data. So we're taking something that's on a range of 0 to 100, and we're turning it into a binary output of saying that, is it a variant or is it not a variant? And when we do that, we lose a lot of information. Well, what if we could, quantify these differences? Instead of saying, well, it's over 10%, why don't we say, well, 24 is closer to 57 than it is to 100. So if someone has 24 and the next one has 57, then they're more likely to be in a transmission chain than the other. So we did that. We took a query genome A and a query genome B, and we looked at all the positions in the genome to look at the abundance of the SNP in question. And for each comparison, we looked at the difference between them, and then we took the mean of that difference. And when we applied this across the entire data set, what we saw was, a bit of a better correlation. So here, these dark purple ones, these ones represent comparisons where it's a direct transmission. This one is like 2 steps away, 3 steps away, 4 steps away, 5 steps away, all the way down to 20 steps away. And you can see the effect sort of tapers down. But this gave us some hope that, and when we look at the mean here, what you see is quite a nice trend. Now, my colleague, Mary Bushman, she's actually come up with a smarter way to do this, which is instead of looking at the average, she's used the square sum, which is, gives a much better correlation. And it also eliminates the having to deal with the negatives. So we said, well, can we model this? Can we put this in a model that gives us some metric of transmission likelihood? So we know what the probability that distribution is for transmission pairs. We know what the probability distribution is for the whole data set. So again, can we apply some sort of a Bayes theorem and fit this data to a truncated normal distribution to get an idea of what the likelihood is? And the answer is yes, it does actually, it's not a perfect model, but it does a decent job of giving you a good idea of who's infected, who's infecting whom. So if we look at a strain here, now bear in mind, this strain that we're looking at here has about 100 other strains that are 0 SNP distance. When we look at them the conventional way, they're identical. But when we look at it in the context of what's happening within the host, what we see is that actually, these strains that are from the same phylogenetic tree, from the same transmission, and are like 1 or 2 steps, are actually more likely to be linked by transmission based on the metric that we've computed. If we go further down that transmission step and we go to, this where everything else that's identical to it essentially has this from the same phylogenetic, same transmission tree, again, you can see a very nice correlation. But then, you do run into some problems every now and then. So here, for example, again, nice correlation. But when you go further out and you pick out something that's here, which has, again, about 30 things that it's identical to, it's a bit more challenging. But this is expected, because what's happened in this case is that this strain has just acquired a mutation. So basically, out of the 2 or 3 strains that are competing, one of them has just won, and it's swept through, and it's now at 100%. So you don't have, for about 2 or 3 transmission steps, you don't have any within host variation. So there's nothing like on top of the SNP difference, there's nothing that can really distinguish these strains. And that's, I guess, something that, biologically you are going to have. But nonetheless, when we look at how well our model does in terms of predicting transmission, so if we just focus on the top hit, then less than 50% of the time, the top hit is actually a transmission pair. But if we look at whether the top hit was within the same transmission cluster, meaning that less than 5 transmission steps apart, at least 80% of the time they were. But also, if we look at the model, what we find is that 80% of the time, within the top 10 hits, we actually get the transmission pair. So you're going from comparing hundreds of comparisons to now just focusing on about 10, 20 comparisons, which is useful for a public health official if you're trying to really get to the bottom of it. And when you look at how, how do you call it? This curve here is just basically showing, so for one transmission step at this point, you have about just under 50% of the time it's your top hit, and about 55% of the time is either the number 1 or the number 2 hit, or 2 transmission steps away, or 3 transmission steps, or 4 transmission steps. And when you have more diversity within your host, that increment is even better. So it's definitely encouraging signs that we can indeed reconstruct chains of transmission when we leverage within host diversity. So in terms of this work specifically, I just want to thank our colleagues in the Hanich lab who've all contributed to this work, as well as our partners at the University of Auckland, Susie, Hannah, and Priyali. And just looking at the time, I think it's a nice time to just touch on the take-home messages. So really, I hope, having listened to this talk, you would appreciate that, transmission studies have a wide breadth, and there's lots of exciting questions that you can ask using a range of methods. I've only touched on genomics, some genomics approaches. You can use modeling, you can use epi-studies, and you can use combinations of these. And there's a lot of opportunities for interdisciplinary collaboration, either with clinicians or with modelers or with statisticians, or even in the field, like we did with our colleagues who were anthropologists. And even when you have samples that are not, even if they're not appropriately sampled, if they're sampled for a different purpose, it can still be informative in terms of transmission. And finally, that, we're getting closer and closer to high precision contact tracing, where we're going to get better and better at identifying who infected whom. And there's also opportunities for modeling things like this. When you have trends of diversity that are correlated with number of transmission steps, you can get to a position where you can now even account for individuals who are not sampled within the population. So on that note, I'll say thank you for coming and for listening, and I'm happy to take questions.
Thanks, Madikay. Very interesting talk. Folks, please raise your hand or put questions in the chat, the Q&A. Just to kick things off, I'll ask a question that comes to mind. I mean, really fascinating here from a research point of view to see what you've come up with. Also, I think what to me seemed like an obvious question is whether or not we could make any use of this sort of technique in a public health approach. And sort of, can you comment on what might make that feasible? Because certainly doing large-scale deep sequencing of individuals for contact tracing in a breakout situations, probably not feasible. Is there anything that you could imagine that might work at scale?
Thanks, Harry. That's actually very much in line with my thinking process, is that really the next step is to think about how we can use this in a public health setting. And I agree that, yes, it's not feasible to do deep sequencing routinely, but we could explore what happens when you downsample. So if you, for example, if we'd sequenced these genomes to 100x coverage, as opposed to close to 1000x coverage, would we still be able to observe these trends? And if the answer is yes, then that makes it closer towards something that you can implement in a public health setting.
Yeah, I mean, the other thing that occurred to me would be whether more traditional epidemiologic contact tracing might be used to give some insights into who should be sequenced. Does that seem like something you might better pursue?
Absolutely, I agree. I think that would definitely make it more, it'll give you more bang for your buck if you were actually focusing on individuals who are likely to be part of a cluster, for example.
Mm-hmm. So since I'm not seeing other questions, I'll just keep firing away here. But I mean, obviously what you've done here, it looks like it's all, it's bacterial. I mean, is there any obstacle to applying any of this to viral breakouts such as COVID or?
I wouldn't say no, because I have seen that I've seen groups try to use similar models for COVID.
Mm-hmm.
And I think it could work. Viruses tend to be a bit more noisy than bacteria, but I think it could work. I have seen individuals look at within host diversity in COVID, and I think it should be able to work. I think there's someone raised his hand. I think it's Wuri.
I didn't see their hand raised, but okay. Yep, let's see here. Yeah, Wuri, please go ahead.
Hi, this is Wuri Jalo. Thank you, Madikay, for this excellent talk. I have more of a comment than a question. Just to add that Madikay is one of my colleagues. I'm also a postdoc currently at the Genome Genetics Center in New York. So I have a similar journey with Madikay. So I'm really impressed by his achievements at this point. Well done, Madikay, and thank you for this brilliant talk. My question is that what is the role? Do you have looked at the role of host or the individual genetics in terms of how this transmission will occur with the bacterial or the pathogen genome?
Right. So thank you, Wuri. And that is a very relevant question. But in this case, because it was a mouse model, they used the mice are sort of standardized in terms of their genetic background, so that it doesn't affect the outcome of the studies. So we wouldn't be able to look at individual host dynamics, or we'd assume that they'd be constant. We know that they could have different immune responses, but we didn't measure, for example, CD4 counts, or we didn't do any immunological markers on the mice that was done concurrently with the with the bacteria to see how it impacted evolution. But it could certainly. We know that selection pressures within the host definitely affect how much transmission you get, or how well a bacteria is carried, or how it can transmit, or the viral load and things like that.
Thank you.
Okay, we also have a slightly cryptic comment in the chat from Emma Fairbanks, who says parasites ? And I'm not quite sure what is meant by that, but Emma, if you want to elaborate on your question, that would be great. Emma, please go ahead if you want to tell us what your question is. Okay, perhaps not.
Yeah.
Oh,
Well, I
hello,
guess,
can you hear me now? Sorry.
Yes, we can. Thank you.
I just, because you were originally discussing that it could be applied to viruses like COVID, and you said it might be more tricky than bacteria, but I was wondering with how easy parasites would be on that kind of scale.
right. I know parasites are a bit more complicated in some cases. So for example, with malaria, we know that there is a sexual reproduction stage in malaria where you get recombination and essentially the genomes get jumbled up. We do have recombination in bacteria, but not quite on that scale where at basically every life cycle you have recombination. And in many cases, when we do phylogenetic analyses for bacteria, we're able to exclude those recombination regions. So I'm not very well versed with parasite genomics, but the little I know would suggest that doing this kind of head-to-head genomic comparison for parasites might be a bit more challenging because of that angle of sexual reproduction and recombination.
Okay, thank you.
Okay, so there's another question here that I'll just read out from the chat. Since this is something you would or may want to apply to a public health intervention, would you still consider genetic aspect of host resistance, for example?
That would be more challenging. And so one of the key considerations that you would make for deploying something in public health is, cost and feasibility, right? And sequencing in itself is expensive enough. But sequencing the host is even more expensive. The human genome costs a ton. So, and I'm not going to pretend like I'm an immunologist, but I would imagine that trying to account for that might be more challenging. I mean, you could model it in, for example, you could model in a situation where, let's say if you're looking at a vaccine preventable disease, you could model in the effect of vaccination, for example. And if an individual is vaccinated, you can take that into consideration. So, but yeah, it would not be trivial to incorporate it. It would not be trivial. It's possible, but it would not be trivial.
Yeah, one presumes that the incorporation of the host resistance would be easier to accommodate or account for if you had some hypothesis as to which genetic factors in the host you're looking for, right? If you're just looking for something, just to see if there's any signal at all, it would be very tough. If you're looking for specific signals, perhaps it would be more tractable.
Exactly,
yeah. Interesting stuff. So I think with that, we're at about the 1 o'clock hour, which is sort of means we've used up our time here. Madikay, thank you for a really interesting presentation and a perspective that's a little bit different from what we usually end up hearing in these talks at MIDAS. And so for that, I think you've expanded our horizons a bit, and we really appreciate it. And thanks again, and we're looking forward to hearing what you come up with in the future.
Thank you so much, Harry. Thanks for inviting me, and thank you to the audience, everyone that came to listen.
Our pleasure, everyone. Remember, thanks, and please join us again on November 19th, 1 p.m., where we're going to hear from a couple of folks from the RAND Corporation. Thanks a lot. Take care.
Thank you.